Hi guys, and welcome again. In this video, I'm going to be talking about interactome and protein-protein interaction analysis. So if that is something that you're interested in, please stay. So let's start from the beginning. I want to answer a couple of questions here, like what is interactome? Why would anybody do interactome analysis? What can we get from it? How long does it take? What are some pitfalls? I want to talk about all of this stuff today with you, so bear with me. The reason why I want to talk about this specific topic today is because I see a lot of people struggling to do their interactome analysis and experiments. Some of these mistakes can be avoided, so I just wanted to make sure that I can put all the information out there and make sure that you understand what you have to do. So we all know that proteins in our cell have different but very specific and very important roles. Majority of cellular processes require that proteins specifically recognize other molecules. Those molecules could be other proteins, DNA, RNA, small molecules, and these are called interacting partners. So that is what we study. Protein-protein interaction analysis is something I spend a lot of time investigating, optimizing, you name it. So this is kind of my passion. This is what I like to do. This is what I think it's really important. So another important thing to mention is that our cells, they change all the time. And the good thing is that we can track those changes. We have this beautiful method called mass spectrometry and proteomics. So using those tools that we have, we can track the changes within our cell. So before going any further, let me tell you what is interactome. So interactome is the whole set of physical interactions between molecules. So in this case, it's going to be protein-protein interactions. So in the simplest word, these proteins form a network. The network of proteins can be looked on a big scale, meaning within the whole cell, or we can target specific pathways and look into how that network changes during different phases, metabolic processes, different pathological processes. And so here is an example of protein network changes due to different glucose treatment of pancreatic islet cell line. So this was a primary cell line. And what was done here is immunoprecipitation of FANK-A protein. So this is Fanconi anemia protein. So with the co-immunoprecipitation, targeting a known protein with a specific antibody can pull the entire protein complex. So not only the unknown members of the complex may be identified, but also all the other direct and indirect interactions. As you can see here, nodes describe the number of proteins and edges are actual interactions. This network was done using Cytoscape. You can observe how dramatically this network changes in different conditions. So let's look at the raw data file. This is a chromatogram of a sample with a low or normal glucose level treatment. And then this one is whenever we treat it with a 20 millimolar glucose, which is a high glucose. Look how big the difference is in these chromatograms. So it's not a surprise that we see such a big change in a network. The next cool step would be to do functional analysis. So this is our paper that was published in PLOS One back in 2019. So I'm going to tell you what is the most important thing to think about before designing your interactive experiment. Well, actually, there are two things. So first thing is to have a good negative control. Good negative control means that you're going to have an IgG, the same species as the antibodies you're going to use for your specific pull down, and then when you do the blot before mass spec, your lane has to be completely clean. And sometimes this takes a lot of time. For some folks that are fortunate, this negative control just works, and you don't see any specific, non-specific bands. It's just clean. For some other people, I see a lot of people struggling to optimize the experiment. So you have to work your conditions until your negative control is really negative. That means no bands at all, especially not having a band on your specific molecular weight where your protein of interest is a good sign. So let's see here how that exactly looks on the Western blot. So pay attention. My IgG is completely blank, so that's a good sign. So my friendly suggestion to you would be, if when you're doing Western blot experiment to confirm that your co-IP works, have an input, unbound and bound fractions all together. That way you can easily troubleshoot if something doesn't make sense later, and you can always come back and see how your Western blot look like. So something to think about. So negative control really has to be negative, meaning clean, nothing there to be seen. Another important thing is having a good antibody that are going to capture your protein of interest called bait. So if those antibodies are not good, your protein will not be pulled down from the sample, meaning that you're not going to be so happy as you will not get the results you want. So during my postdoc, I spent a lot of time, and I mean a lot of time, optimizing my experiment for interactome analysis. So I'm going to share what were my best conditions, the most optimal conditions. And the good thing is that now when I work at a mass spectrometry core and I'm dealing with a lot of interactome analysis from other researchers, those conditions kind of work for majority of you guys. So I'm going to share the table with these values. So what you're going to do here is start small scale, meaning with the Western blot. So you need to confirm that you can specifically pull down your protein. You will start with the Dynabeads. So Dynabeads are awesome in a mass spectrometry field because they're just much cleaner than other beads. You will need your specific antibody, of course, and then your protein from the protein lysate. You're going to start with the 1 milligram of Dynabeads, 5 micrograms of antibody, and 250 micrograms of the total proteins. If you can successfully show your pull down, it works, then you are going to start thinking about the large scale mass spectrometry. And basically what you want to do here is tenfold your values from the Western blot. I think the worst thing that you can possibly do is not using enough antibodies. So if the protocol says 50 micrograms and you're like, let's try 10 or 15 or 20, guess what? It's not going to work. I promise you it's not going to work. So 15 versus 50, let's see what happens. This is the spectra and the chromatogram with the conditions when 15 micrograms of the antibodies were used. Somebody was trying to save some antibodies, but unfortunately the results were just really bad. This is when we used 50 micrograms of the antibody, and you can see that the spectra and the chromatogram looks much different. It's still not perfect, but you see my point. So you will end up using all of your time, all of my time, then, all these reagents cost, right? So it's going to be a money loss. Experiments on the mass spectrometers are not cheap. So you want to make sure that you have all the conditions right. That means your pull down works. You use enough antibodies. Really important. Don't be cheap. Use those antibodies. Your negative control is negative, means clean. Before you do anything, big scale, large scale, please optimize the conditions, do the Western blot. If it looks beautiful, it has to look beautiful. Then you're ready for the mass spec step. Also, another important thing is, what is your sample? If your sample is a, let's say, tissue, cell line, you're probably good. If your sample contains blood, then you're going to have problems. You know why? Because there's going to be a ton of sticky proteins, really abundant proteins that is usually not the protein of your interest, like albumin. Even if you deplete it it's still a lot. So try to do something about your sample before you do anything mass spectrometry. That means albumin has to go. You have to try and find a way to remove those things from your sample that are potentially going to make problems, because you know what, usually the protein we look at is not the most abundant. It's just like a tiny amount, small amount in your sample, really valuable. So to be able to see it and recognize it and have peaks from that protein, you have to remove all those sticky things from your sample. So if you don't know what to do, it is a good idea to talk to somebody with more experience. Usually people from the proteomics course can help you and guide you. We certainly can. So please mind that thing. It's really important. So at our mass spectrometry and proteomics core facility at the University of Nebraska Medical Center, some of our protein-protein interaction services include co-immunoprecipitation, LC-MS/MS, data analysis, network generation, pathway enrichment analysis, as well as troubleshooting and consultations. So if you have any questions, feel free to contact me either using my email address provided here or on this YouTube channel, and I will be happy to help you. Thank you for watching.